Now, our next speaker is Jorge Bernadino de la Serna from Imperial College London, who's going to talk to us about imaging cholesterol.
Well, I will try to be brief. I guess you're probably thinking on your lunch already. And I'm going to change a little bit gears because we're going to talk a little bit about lipids and in particular cholesterol. So I think probably I need to use this if I want to, right? So what is so special about cholesterol, right? So, well, cholesterol is this molecule. So we've seen very complex molecules, and this is actually quite small and special molecule. Only this part is polar. This area here is a polar. This is rigid. However, it has a tremendous importance. It can go in the membrane everywhere, and it can change many biophysical properties of the cells, right? So it can be a structural molecule because it can change the fluidity and the tension at the plasma membrane. It can change the permeability of the cells, allowing communication, for example. It can change the plasticity and the mechanical coherence of the cells. It's also a regulator because it can act as an island or platform where some proteins and receptors are happy to be with, and they can interact with lipids. And of course, they can form signaling hubs. Moreover, it's a metabolic molecule and is the backbone of many steroid hormones and vitamin D analogs. So, for example, all these events, let's see if this works, by the way. Yeah, all these events will probably not happen without cholesterol. Right? And these cells are basically doing what they normally do, right? They divide, they communicate, they touch each other, right? Without cholesterol, this will be impossible. I'm going to focus initially on cholesterol in the plasma membrane as signaling platforms, which you probably have heard already quite a bit. And I'm going to try to, at the end of the talk, actually come with all these numbers, which actually are quite new. And I'm actually quite happy that I was able to be involved in several projects that would allow us to give us all these numbers, right? And most importantly, the signaling platforms that where cholesterol is part of, actually they play a good important role from mechanics in membranes and permeability to actually gene transcription. I have to do a disclaimer at this point. So the R-word draft, most of you probably heard of, and is quite popular. So I have to say that during the talk, I'm not going to talk about this word. I'm going to talk about phases, liquid order, liquid disorder phases, because it's what I am actually more familiar to. And the main reason is because, especially after convenium in 2006, the definition of RAF make our lives for biophysics very impossible, because basically it told us that a RAF has to be composed of saturated lipids, cholesterol, and sphingomyelin. It has to have particular biophysical properties, meaning particular fluidity. It has to have a very tiny scale, spatial scale, so they are nanometric, and they are very fast, they are transient, right? And even though inside those domains that apparently that order, the molecules have to move. In my opinion, all these components are impossible to pinpoint in one single experiment. So honestly, if I will have to define something as a myth, I will actually probably use these terms, right? It's impossible, right? However, it helps us to bring everybody in the same sort of platform and grounds, right? So actually, interestingly enough, I'm going to start talking about platforms or signaling platforms in the primary cilium genesis, right? How are these, the primary cilia is generated, right? And this actually was one of the initial questions that actually Kai Simmons started with, and where actually he was interested with RAFs, right? So again, I'm going to see if this video works, right? I would like you to have a look. This is just fluorescent intensity, plasma membrane apical site of an epithelial polarized cell. These are actually cilia baiting, right? I'll see if I can put it again. Might be not possible. Yeah. Okay. Hopefully, no. Yes. Okay. So these are baiting cilia, but I would like to draw your attention to this particular funny element that we see here, because this is actually where the primary cilia actually stems, right? So, and this is what I'm going to focus the talk. And I'm going to try to convince you that I've been able to resolve the molecules and the different elements that has been involved laterally with the lipids to try to generate the primary cilium. So to start with, classically, when we wanted to look at lipid in membranes, we normally look at fluidity, right? We wanted to look at these signaling platforms because they are highly enriched in lipid saturation, so saturated lipids, sphingomyelin, and cholesterol. So they are basically quite rich, right? So what we will have called probably liquid order. So this would be an O, liquid order, right? So a very nice way to look at that is using fluorescent microscopy and employing subattochromic dyes or polarity sensitive dyes. They actually stay here. They sense the water content around, basically in the environment. And depending on that amount of water, they emit differently in the spectrum. So when you do a ratio between these two emissions, you subtract the emission between one another, and then you normalize it, you get something that we call the GP, which is the general polarization. And when the GP is above 0, we consider the membrane is order, right? It's highly order. And when the GP is below 0, it's highly disorder, right? So this is not absolute numbers, but it's a very nice way to characterize your membrane, right? In living cells. For example, in the primary cilium, for those that actually know a little bit about this, they start, well, I'm going to talk in particular about the non-canonical primary cilium generation, which is quite understudied. And this actually doesn't contain the ciliary pocket, right? So it actually emerged from the division, right? And this bit here actually is part of the Fleming body, right? So when two cells divide, one of them actually gathers that Fleming body or mid-body in the remnant for some other people. And this, when it starts in the periphery, then actually moves towards the center when it starts to generate the primary cilium. When you characterize the fluidity by looking at these properties I just told you, you actually see that when you see red from now on in these maps, these fluidity maps, it will mean that the order is very high. So the lipids are very close together, highly packed, right? When you will see blue or green, they are less packed, they are more fluid, right? So clearly, where the ciliary is going to stem or when it's going to generate is actually quite ordered, something that was already known. But it was quite interesting for us because we actually developed methods to try to push the technology further to understand even more things at the molecular level. So this is a marker for a molecule that actually is coming from the mid-body remnant. So we actually know that this actually is coming from there, right? Another interesting part or another way, classical way for biophysicists to look at the membrane is to look at the diffusion of the lipids, right? So because if you're looking at the northern membrane, the lipids will diffuse laterally slow, right? If you're looking at a liquid or disordered membrane, more fluid, the lipids will diffuse laterally faster, right? So that's another way. It's again, not absolute, it's relative, but it gives you a lot of information, right? Indirect information. So there are many different ways. A classical one is to use fluorescent correlation spectroscopy methods. In this case, I'm going to show you laser scanning, right? So instead of just going to obtain a point, a focal point, and obtain the diffusion of the lipids in that particular point, what you actually do is draw a line with your laser, and then each of these pixels, actually, you will be able to obtain the diffusion of the lipids in each of these pixels, right? So what you can do is just basically draw or graph the autocorrelation function of these lipids moving in that focal spot, and it will give you something as nice or ugly as you would like to say as this, right? But this is biology. And when you actually separate each of these lines in this carpet, it will tell you which is the diffusion of each of the molecules, in this case, the lipids that you're looking at each individual pixel, right? So you know now how fast the lipids are moving in each individual pixel. You will see this is actually living cell. So we'll see that's why you have these spikes. So some lipids are moving faster than others, right? So midway of this decay curve, if you want to call it, is sort of what we call the lag time or the transit time, sorry. And the transit time is the amount of time that a certain molecule has spent in that focal volume. In this case, it's a lipid, but it could be a protein, okay? So this is the way, classical ways. Either you look at fluidity or you look at indirectly fluidity by looking at the diffusion, right? So let's come to the biology. and let's see how actually the primary cilium stem. And when we started to look at this, we actually use classical fluidity measurements with Laudon. So we'll look at the GP. As I told you, whenever you see red, it will be highly ordered. When you see green or yellow, it will be less ordered, right? So, but interestingly, when the midpoint remnant was traveling towards the middle, it actually start to split, right? And actually we have here centering, which actually where the primary cilium will stem, right? So suddenly, I can tell you this is our part of the midpoint remnant, and suddenly it's splitting, right, in two, and is generating a very highly condensed membrane where the primary cilium is going to stem, right? And I can prove you that that actually happens because in 3D, you can see how they separate and how the primary cilium start to grow all the way, right? And again, everything is highly ordered, right? And we still have the centering here. So what we actually found is that, and actually we name it this way, we found that there is a remnant associated patch, right? That splits and forms what we call a centrosome associated membrane patch, which is this one, right? And from here is what the primary cilium stems. So we try to characterize all this. I have already told you that it's actually very important to look at diffusion, especially with lipids, right? And I'm actually quite interested in live cell imaging. So what we did here, if you remember, this is exactly the previous video I just saw you, but in this case, this is actually a fluidity map. So again, when you see red, you will see high order, or you will see green, actually, yeah, blue, it will be low order, right? So I'm going to play the video again, hopefully it will work. And you will see exactly the same, but I would like to draw your attention here again, right? So this is the normal apical part of the cell, and this is where the primary cilium is going to stem. I hope you have been able to realize that there was a gamut of colors, from red to yellow to blue, right? So that means that in that area where the primary cilium is going to stem, that we before thought it was completely ordered, suddenly there is more than one kind of phase. So actually there are order and disorder, liquid disordered phases. So we actually try to get more information about this. And then we actually look at the technique that was developed in 2005 that is called RIX, that is raster imaging correlation spectroscopy, right? And then we combine this with polarity sensitive dyes. It's basically what I would call an elegant trick. An old technique using dyes that are also old, but nobody has actually mixed them together. And suddenly what we found is that we can get a lot of information. We can get spatial information, temporal information, we can get the dynamics, and we can also get the ordering. So we are not now needing to do one thing or the other. So in one single technique, we can get both things, right? So RIX basically does is rastering, and you just need to do autocorrelation between one pixel and the next one, and then you need to do autocorrelation between one line and the next one. Because you're doing this in a full frame, then allows you to have a much larger field of view, and suddenly you obtain information of much larger sort of areas within a cell. So it's not just one focal spot, small focal spot. You now have a very large field of view. So when you analyze that little spot here, you actually see this. So you can actually quantify the map of ordering within the ring. And right, so sorry, this is actually not the map. This is the map of ordering. So you will see the GP with higher order areas and lower order areas, right? And you can also at the same time quantify the diffusion of the lipids within this area, which is 0.43 micrometers squared per second. You can, of course, obtain how many pixels are with high order, how many pixels are with low order, right? Interestingly, because you have a very large region of interest, suddenly you can obtain information in ROIs, regions of interest, right? So I can focus on this one, but nicely enough, I already have my control there because I have a large cell that actually is not expressing, is not generating that primary cilium base. So this is actually how the plasma membrane is actually happening, right? It's actually behaving. So the diffusion is at 2 micrometers squared per second. And as you can see, the GP is mostly fluid, right? So this technology has another advantage. So this dye, the one that we use is not anymore Laudon, it's a NEP. And a NEP actually allows us to use a STED on it. So we can actually obtain super resolution on this dye. So when you look at this sort of base of the primary cilium, I hope you can see that there are sort of two colors merged, right? So again, that is the two emission intensities from this dye, and making a ratio of those will give me again that information about fluidity or less fluidity, right? So I hope you will see that actually you get a much nicer interested image when you apply a STED, right? So again, you can quantify the different order within the areas, right? And you can quantify how many pixels there are in order state or in less order state. Interestingly, I hope you see there's these small sort of protrusions, which are actually going to be quite interesting, because these protrusions are being released and removed from the base of the primary cilium along the primary cilium is forming. So the primary cilium is refining, generating the primary cilium, but is getting rid of precisely those areas of lipids that are more fluid, right? So because you can do STED, on top of that, what we define is, or design is a new experiment in which we can actually apply the interesting concept where STED can give you dynamics as well. So we actually developed this method that is basically interleaving confocal and STED, right, at the same time, and we do rigs, right? We obtain the analysis of rigs by first putting confocal sort of normal excitation and then the STED super resolution. So when you do that, the nice thing is at the same exact time, you get the information in super resolution and you can compare with diffraction limited. Why is this so important? Because the lipids normally, when they try to diffuse, they normally find they are hindered normally by either cytoskeleton. You probably have heard of the picket fence sort of a diffusion model. So a way to understand if the lipids are really being hindered, stopped, right, in the diffusion, actually is by applying a STED, because a STED allows you to have a look at much smaller focal points, right? So when you do this, you can actually get the diffusion in confocal and the diffusion in STED. If you do a ratio between the STED and the confocal, directly you get if the lipids are moving freely, they are doing free diffusion, or if they are actually hindered, right? So if you look at this in the plasma membrane, most of the lipids are freely diffusing, as one would probably expect. When you look at this at the base of the primary cilium, most of them are actually hindered and very little of them are actually free diffusing. So with this, there is actually no doubt that we have a method, a technique where we can actually know how fast the lipid moves, where they actually are, and whether they are hindered or not by proteins that are around them. Okay. So we actually tried to push this method further. And what we did is, because you're actually taking a snapshot, basically you're doing a video, right? That allows you to have also further information. You have the time information, right? So you now can actually analyze different time points. So you can actually analyze the first 50 frames, which basically are the first 50 seconds of this event, and then compare to the next 50. And because this is a dynamic event and it's actually a new generation of membranes, this is actually quite interesting, right? So you can do that by post-processing with exactly the same experiment. And you can just analyze the regions of interest within the base of the primary cilium that you have interest. And then you can obtain this, what we call DRAT. DRAT is basically that will tell you, again, if there is close to 1, it's free diffusion. If it's below than 1, it will be hindered diffusion, meaning that there is either proteins or just the lipids are moving slower, right? Quite interestingly, if you look at here, most of these areas, even with high intensity here, they show you free diffusion mostly. And that was actually quite surprising for us. Why actually lipids will, in this area that we have think and we have seen that is mostly rigid, why they actually do freely diffuse, right? So what we could do, again, is another layer of information. You can apply another technique that is called number and brightness. And number and brightness is looking at the molecular brightness of your fluorophore. And that gives you actually the stoichiometry of the events. In the case of the lipids, it's very difficult. I will give you how many lipids you have in a particular area, right? But it will actually give you the collective assembly map of the lipids. So how actually the lipids move, diffuse collectively, right? So when you do that over time, you actually see that over time, the primary cilium, where the base is actually being refined. You can see that it's smaller and smaller, the size is becoming actually more rigid, right? And over time, what you get is actually more collective assembly of lipids with higher degree of order in the center. So this technology actually allows us to finally conclude or summarize that during the primary cilium formation, what actually happens, and if you look at this very nice time series with the STED, you will see that the membranes, actually this area is quite fluid, but over time actually becomes very rigid. So this actually forms a bottleneck. And what we can see is that these budding structures are being released. And the way they are being released is because there is a high membrane order mismatch, right? And it's a high increase in the line tension. And that's why, of course, with proteins that we were not at this moment looking at the proteins, right? We were only interested in the lipids. They are pinching and being released. So over time, what we see is an increase in the molecular crowding, an increase in the lateral packing of the lipids, and membrane condensation, and a slow dynamics of the lipids, and definitely a concerted assembly dynamics of the lipids to try to form this very tiny, highly condensed membrane patch to generate the lipid. So hopefully I have time to tell you now about cholesterol, because as this was basically across scales. So first we have to look at the domains, right? But in these domains, we were actually not looking at just cholesterol. We were just looking at areas enriching cholesterol. And we know that they're enriching cholesterol because we have, of course, used drugs and we have controls where we have seen that we have high level of cholesterol, right? So what we did actually in collaboration with a person in the Basque Country, in Bilbao, he developed this probe, which is very nice because you can just covalently dye to DARCO, which has different dyes. And this is basically the same as a cholesterol. So we want to test if we could actually, reproduce the same effect and same behavior as cholesterol, right? And we did this in these cells, and we applied STED to it, and hopefully you will see that, and this is the first time I've seen in a living cell that you see patch structures at the membrane. This is something that normally you wouldn't say, even if you put any lipid, if you label any lipid. So this is sort of the first evidence that with a dye, you can actually see sort of islands or patches of cholesterol along the membrane, right? So when you do your analysis of the different sort of a classical intensity line on your STED, we actually found that we can resolve things down to 25 nanometers. And we actually, if you highlight several areas within the cell, I hope you can now see easier the patch, membrane patches, right? So this is confocal, this is STED, confocal STED. Very nice patches of cholesterol domains, right? If we actually analyze all these, we actually found that you have two kinds of domains. You have domains that has a size around of 50 nanometers, 48, right? This is, of course, a distribution. You have even less, right? And the other distribution was around 150, right? So this is actually, for us, the first time that we were able to see different kinds of cholesterol domains. And again, we apply dynamics because cholesterol, again, and the lipids is very important because it changes the fluidity of the membranes. So in this case, we actually apply laser scanning STED, right, FCS, and we again interleaved it with confocal to again get the same information as I just showed you before. So we will be able in one single experiment to detect which areas are free diffusing, where the cholesterol is literally free diffusing, and which areas of cholesterol actually are hindered. And as you can see, most of the population of the cholesterol that we see is hindered, right? So it's in highly ordered domains, and some of them, they are still in free diffusion, freely diffusing. Interesting, again, is that you can quantify from here the molecular brightness of the molecules, and that gives you a further layer of information because now we can tell more or less how many molecules of cholesterol we have in each of the different domains. So actually with this and doing the calculations, we can see in the domains that are very tiny, very small, we found around 17 molecules of cholesterol, and those will be the liquid order domains. And we found around 60 molecules of cholesterol in the liquid disorder domains that we were looking at. So again, you can look at this live, and you will see that the probe, hopefully, you can see now. Okay. Working now? Yeah. So this is again the cells. You can hopefully see that there is patch membrane. There is also a lot of vesicles forming, right? It was actually quite interesting to see that we could quantify the vesicle by doing vesicle tracking. And why we decided to do that? Because we found that the vesicles containing cholesterol, interestingly, many of them, they're actually spending quite a long time at the plasma membrane, and not only being trafficking from one side to another along the cytosol. So these are the tracks, the total tracks. These are the tracks that we find at the plasma membrane, and these are the cytosolic track. So when we isolate the ones at the plasma membrane compared to the ones in the cytosol, we see that the ones at the plasma membrane, the diffuse is lower, they stay longer time at the plasma membrane. The one that are at the cytosol, they actually move faster. So this is rather interesting because then it probably means that the cholesterol vesicles are being attached to the plasma membrane for a particular reason. And we actually were looking at this, and over time, we were able to find how these vesicles actually dock, release cholesterol, and then are removed over time. We can even quantify how much amount of cholesterol we have. And this is the color scale telling you how many cholesterol we have in a confocal movie, right? But of course, we can apply a STED, and this is the plasma membrane, where we can quantify the diffusion of the lipids of the cholesterol in the plasma membrane. We can quantify how many cholesterols we have at different points of the plasma membrane. We can do the same when the vesicle comes in, quantifying the speed at which the lipids move, and we can actually quantify how many cholesterols we have along the plasma membrane and the vesicle. So this is just to show you that a normal membrane will actually just fluctuate the intensity in this way. However, when you have a vesicle, you actually can see when the vesicle moves and joins and docks to the plasma membrane. So hopefully now I can actually comment on all these numbers, because from this technology and all these experiments, we've been able to actually say that our nanodomain shape is most of them circular, that the size goes from 20 to 100 in our, of course, experiments, that there is around 60 molecules of cholesterol in liquid odor and around 17 molecules of cholesterol in liquid disorders, which is diffusion speed of 0.13 micrometer square or 1.26 in the liquid disorders. And of course, we have even quantified how much time more or less the cholesterol stays in a domain, and we've seen that is a little bit more than 4 microseconds. Finally, I would like to thank the people that has been involved in these projects. So with the primary cilium was Professor Miguel Alonso from the Centro de Biología Molecular Severo Ochoa in Madrid, and the person who developed the dye, the cholesterol dye, Xavier Contreras in the Biophysics Institute in the Basque Country, and Esther was a postdoc that was working with me, funded by the European Union, and this is where I am now. Alexia was involved in the cholesterol work, and this is part of the funds that I have, and I'm enjoying now. Thank you very much.
Thank you. We might have time for 1 or 2 quick questions before we break for lunch.
Yeah, very nice talk. How do you factor in the involvement of proteins into these membrane dynamics that you are studying?
In those studies, there is no factoring of proteins involved. In the other ones that I do, I actually do cross correlations. So I put proteins, I have labeled them, and I cross correlate the diffusion of the lipids with the proteins. But this one was mostly about cholesterol and just the diffusion of the lipids in the domains. But the fact that they are retained with the STED and they are hindered, that tells me that there are definitely proteins there.
Okay, I think we should probably stop there. I'll thank all of our speakers again from our session on imaging across scales.